as many know, is a project that aims to map the cells in the tissues and organs of our body. And the website is there, and the aim is to create a comprehensive reference map to further our understanding of our cells in terms of their molecular fingerprints, and also in terms of providing a framework for translational insights into disease diagnostics, regenerative medicine, and so on. The project now goes back about 5 years when Aviv and I got together to arrange the initial kickoff meeting in 2016. And now we're at over 2,000 members and over 1,000 institutes from all over the world. And I encourage everyone to join the Human Cell Atlas community. Very simple, humancellatlas.org/joinhca. And the computational community is really at the heart of this project, as I'm sure you'll see in this talk. The technologies that catalyze the formation of the community are the resolution revolution in genomics that we've lived through over the last decade or so, through single cell genomics, and then the spatial genomics technologies and spatial imaging technologies that allow us to study the tissues in their 2- and 3-dimensional context. And so that's really kind of moved us forwards from the conventional bulk genomics and the lower plex imaging methods. And so it's these technologies that are brought together by computational methods that are allowing us to define the cells in their tissue context and create this Google Maps of the human body. And just to give you a little taste of the data sets, that now number almost 50 million cells, you can see here for the kidney, for instance, the number of cells and the number of individuals that are available, and in published in different databases, and also unpublished work. There's more work coming. And then also, of course, we've lived through the pandemic, and we have studying COVID-19 patient samples, and that's what the little virus means. And you can see the skin, the airways, over almost 3.5 million cells from upper to lower airways, nasal, all the way down to lung parenchyma. The gastrointestinal tract, millions of cells. And human development, which is a really exciting area of the Human Cell Atlas community. And then the liver as an example. To very briefly, so that's really just everything that I want to say, just very briefly about the Human Cell Atlas community. The main message is really, I encourage you to join, to engage. If you sign up, you get the newsletter, the meetings announcements, and so on and so forth. And we just had a general meeting that had over 2,000 attendees from all over the world. And that was really amazing, because the first meeting 5 years ago we were only able to invite about 100 people. And so in a way, the pandemic has allowed us to kind of reach out to the global community and draw people in to both generate data, but collaborate on technology development and so on. So there are many different dimensions. Actually, I'm going to have 3 parts to this talk. And this is bringing me to the second part, which is I want to give you a short taste of what a Cell Atlas project can look like. And the one I'm going to present is our recent work on the human uterus that's on bioRxiv. The link is here. It's unpublished data, but it's posted on bioRxiv. And it's a collaboration with Rosa Venter-Tormo, who leads this project. She started as a postdoc in my group and is now an independent group leader at Sanger, and also collaborators in the university and in Cambridge. And the exciting thing about the uterus is, of course, that it's a dynamic tissue. It changes throughout the menstrual cycle, the monthly cycle, and it's very species specific. As all reproductive tissues, it evolves very quickly, and so even the human versus the primate has differences, for instance, in the structure of the vasculature, the menstruation itself is primate specific. So the mouse doesn't menstruate. It has a diestral cycle, and that means that this has been poorly understood in terms of human biology, because the mouse isn't a very good model, or it's a very accurate model of humans. And so this gave us a lot of scope for new discovery, of course. And so you can see the different phases here, the shedding, the endometrial shedding, which basically means that the upper layer of the tissue is lost. Then the tissue regrows and proliferates, so there's a lot of cell proliferation. And then it also changes its identity or characteristics to secrete nutrients that allow the embryo to implant in the wall and form with the placenta, to form the interface between the placenta and the uterus. And it's that dynamic process that we're studying in terms of understanding the changes in the tissue in this project. And the technologies that we used are cell atlas technology, so single cell and single nuclear RNA sequencing on the one hand, and spatial transcriptomics using the 10x Genomics Visium platform to study tissue sections on the other hand, and then integrate that data with computational methods. And we collected tissue samples. So one of the challenges of studying the human uterus is that tissue samples are not readily accessible in terms of full depth biopsies. So biopsies of the upper 1 or 2 millimeters of tissue are available from diagnostic biopsies where there's a suspicion of a disease, but the tissue ends up being healthy. So we have many of those very thin, small biopsies, and these have also been used in a recent Nature Medicine paper. In this work here, we were fortunate to get access to full thickness biopsies, and you can see here the difference between a biopsy is shown, roughly speaking, schematically by the black bar here on the left-hand side, which is a biopsy from a. We are fortunate in Cambridge to have a collaboration with the transplant surgeons, Korsai Parsi. And from them, we receive full thickness biopsies from deceased organ donors. And you can see here, this black bar on the left-hand side means that we can go right from the inside luminal layer of the endometrium, the tissue, all the way through down to the myometrium, which is the smooth muscle layer that goes around the outside of the uterus. And so this gives us a literally deeper insight into the architecture of the tissue. And so by working with these large biopsies from different stages of the menstrual cycle, you can see we had 7 of these deep biopsies in the first instance. We got another 4 since this slide was made, giving us 11 in total from different phases of the menstrual cycle. And that's an incredibly, although it's a modest number, it's an incredibly valuable collection to have to allow us to then apply these technologies. And so what you can see here is on the top right are tissue sections from the luteal phase, that's the secretory phase, the end of the menstrual cycle. And so this layer up here, this is the inside of the uterus, the cavity, and then down here is the myometrium. So just to give you a sense of orientation. And what's marked in the color scale on this on the right-hand side is the intensity of the gene expression signature of glandular epithelial cells. So these are epithelial cells that form the glands that do the secretions. And you can see that these glands are dotted throughout from the basal layer to the luminal layer of the tissue. They're present in this punctate way. And if you contrast that with this particular fibroblast population, these are the gene expression signature for these fibroblasts is more in the upper layer. And to explain from a computational point of view, what we're using here is a deconvolution approach called cell to location that's also on bioRxiv, developed by Omar Bayraktar and Oliver Stegler with PhD student Vitaly Klepchevnikov. And this is what we use, this probabilistic model is what we use to deconvolute the transcriptome at each voxel of the 10x genomics Visium, which you can see here as circular features. And we deconvoluted each of these multicellular transcriptomes using the single nuclear RNA sequencing data that we had for each of the same biopsies that we got the tissue sections from. And then if we use so we define these cell states from the single nuclear RNA sequencing, so the glandular epithelial cells here, the basal fibroblasts here, and we're then effectively locating the expression signatures of those cell states into the two-dimensional space on the Visium section where each voxel is 50 microns, so that can be 20 to 100 cells. And the basal fibroblasts you see are positioning nicely here in both the follicular and luteal phase. They're present throughout the menstrual cycle, sitting at the junction between the myometrium and the endometrium. These ciliated epithelial cells are sitting in the luminal layer here. And so in this way, we position the cells in the voxels, in the 55 micron voxels of the Visium. Now, this cell to location, I'm just going to go very quickly here with this animation, if it works, is, as I mentioned, developed by Omar Bayraktar and Oliver Stegler's group, and Vitaly made this animation where he's showing the individual cell states being positioned into the Visium data on the mouse brain, so the single nucleus and slide-seq, and here he's showing it for a slide-seq data set to show where the cell states are positioned. And he also applied it to human lymph node using tonsil single-cell RNA sequencing data into the lymph node, showing where the B and T cell cluster in the tonsil, in the germinal centers of the tonsil. And so this is basically showing you how the modalities can be combined. I'm not going to go into the cell to location in detail. It's on bioRxiv. You can read it there. The new discoveries from this was that we showed that there are new epithelial progenitor states. They're not they're different from the types of cells that are in mouse glands, and these progenitors give rise to we think both the glandular epithelial cells and the ciliated epithelial cells, so the two different kinds of epithelial cells. And you can see their positioning here is very different layer, whereas these are an epithelial, a proliferating intermediate in orange is in a higher layer, and then a SOX9 LGR5, so this is Wnt receptor are also in blue here in the upper layer, and this is so these are new insights into the sort of adult stem cell progenitor compartment of these epithelial cells, and I'll show why that's important to understand from a disease point of view later. In terms of resolving the single cells, with the single molecules even, at higher resolution, we need to go to microscopy because the spatial transcriptomics is giving us a kind of coarse-grained landscape. But if we want to see individual cells or a subcellular resolution, individual molecules, we need to use multiplex FISH. And Kenny Roberts did these experiments in Omar Bayraktar's group to show where the EpCAM marking epithelial cells, MMP7 matrix metalloprotease, and this PAEP, which is expressed by the glandular secretory cells, are located. And you can see these different temporal dynamics shows the change in tissue architecture as you go through the menstrual cycle, and you see the glands forming during the proliferative phase and are intact here in secretory and mid-secretory, and then break down at the end. And you can see this here in beautiful high resolution, at subcellular resolution in the imaging. Now, if we want to basically understand what are the transcriptional programs that are controlling the differentiation of those two epithelial lineages that I mentioned, so the glandular on the one hand and the ciliated on the other hand, we use Dorothea, which is developed by Julio Saez Rodriguez groups, and it's Luz Garcia Alonso from Roser's group who who spearheaded all the computational analysis, and she showed that in the ciliated lineage, we have FOXJ1 has 6, which are key transcription. So the expression module is linked to the Wnt signaling pathway. And on the other hand, in the glandular epithelial cell lineage, we have transcription factor CSNRP1, FOXO1 that are linked to Wnt inhibition, and other transcription factors that are linked to Wnt activation, to Notch activation, sorry, HEY1 and HES4. So the transcriptional signatures are giving us clues as to the different signaling pathways that are instructing these 2 different epithelial lineages. And where do those signals come from? The Wnt and the Notch, where are the cells getting these signals from? If we look at the expression across the Visium chip, we can see that there's Wnt signaling in the lumen, in the luminal, so in yellow up here, and in Wnt inhibition in dark blue, Wnt inhibition signals like axin-1 in the kind of functional layer in the middle of the endometrium, and then in the basal layer down here in red and orange, we've got the Notch activation higher, and we can validate that by staining using FISH. Now, where are those signals coming from? The Wnt activation, Wnt inhibition, and Notch signaling. To track that down in the tissue microenvironments, we could, and to pinpoint the cells that are secreting these signals and sending the signals to the epithelial cells, we use Cellphone DB version 3.0, which is an extension of our cell-cell communication database published a couple of years ago in when we studied the placenta and the decidua. And this is Roser's work that was published in Nature in 2018. Luz Garcia Alonso now extended this statistical framework to identify not only the differentially expressed ligands and receptors on complementary cell states, but also whether they are in close proximity in the same microenvironment in the spatial transcriptomics chip, and using those different pieces of information to infer high confidence cell-cell interactions. And that gives us basically the clue that there are multimeric complexes that are receiving Wnt signals, either activation or inhibition, and Notch signals in the different from the different fibroblast populations. Which is a kind of structural cell, is important in terms of making microenvironments that foster epithelial cell differentiation and make glandular epithelial cells or ciliated epithelial cells that line the inside of the endometrium, the lumen. And so we have these in the glands, we've got Notch high, so Notch 2 secretion by the fibroblast, but Wnt low, whereas in the interior of the endometrium, we've got Wnt high, Wnt secretion by the fibroblast that's sending signals to make ciliated cells. And so this I wanted to give you this as a kind of framework to basically walk you through a human cell atlas project and the different genomics and computational elements that are needed to build a map of the tissue like that and understand what are the cell states and how are they talking to each other. And I've acknowledged Rosa Bento-Tormo, who's the rising star and junior group leader who led this work, Luz Garcia Alonso and Louis-Francois Hanfield, who worked together on the computational side with other members of Rosa's lab, Christoph Polanski from my lab, I've mentioned Kenny Roberts and Vitaly Klevchevnikov from Omar's lab, the support teams at Sanger, Margareta Turco, Ashley Moffat, collaborators in Newcastle, and of course the donors and the funders. Now I'm going to switch gears and focus for the remainder of the talk on MultiMAP and the computational method for integration of multimodal data. And before I go any further, I just want to acknowledge two key people, Mirjana Efremova, who was a postdoc in my group, who's now an independent group leader at Queen Mary, and Mika Sarkin-Jain, who's a PhD student in the group now. He did his master's with me, co-supervised with Austin Lamacraft in the physics department in Cambridge, where I have an appointment. And also thanks to other collaborators and other members of the group for different elements of this work. Now, in the previous section of the talk on the uterus, I presented single-cell RNA sequencing and spatial transcriptomics. Of course, you'll be aware that there are other modalities from measuring molecular features of cells at single-cell resolution, including chromatin accessibility, so single-cell, single-nuclear ATAC sequencing, and obviously the transcriptomics, but also protein abundance with site sequencing. And in fact, now we can measure some of these things at the same time, but more frequently we have separate data sets for these different modalities, and we want to integrate them to achieve the kind of integrated picture of the phenotype of the cell like I presented in the previous section. And achieving that integration allows information transfer from one modality to the other, that allows more robust and deep analyses and joint analyses, and it can reveal features of a cell, for instance, its spatial location or chromatin regulatory features that you don't get when you consider just the transcriptome on its own, for instance, or just the spatial data on their own. And so there are different computational approaches to achieve this. Transfer learning from one modality to the next. Matrix factorization on shared features, for instance, is another one, and correlative approaches. And what these approaches achieve are a more holistic view of cell and tissue phenotypes, and also integration for downstream analyses like those differentiation pathways that I talked about towards glandular and ciliated cells, joint clustering, regulatory network inference, and so on and so forth. Now, there are different multi-omic integration approaches that are out there. Seurat, Liger-Connors, Gluer are some of them. And the one that I'm going to present in this talk now is a new one called MultiMAP, which we've have an old version on bioRxiv, and also the code is on GitHub. Now the different approaches have different strengths and weaknesses. The key feature, I would say, from MultiMAP is that it allows you to take into account features that aren't shared between data sets, and it also allows you to integrate multiple data sets. It's non-linear and it scales well. And I would say this scaling is something that as a computational community, we really do have to be aware of going forwards, because the data is just growing exponentially, and a method that works on a modest data set just kind of breaks when you apply it to larger data. And so MultiMAP basically ticks a lot of these boxes and rises to a lot of these challenges. And in particular, they're taking into account features that aren't present in different modalities is also important. And I'll just use that to take the example of memory T cells, where our T cells harbor the memory of infection in the epigenome. And basically in memory T cells, only very, very small changes are stored in the transcriptome, but a lot of the changes are imprinted in the epigenome, in enhancers and so on. So the naive versus the memory T cell has a lot of differences in the epigenome, but only very small differences in the transcriptome. And so being able to have an integration between transcriptome and epigenome that takes into account ATAC sequencing peaks is very powerful, and MultiMAP is the only one that allows that. So the principle of MultiMAP is that you basically find one shared multimodal manifold for all the different modalities. So you can have multiple data sets from the same modality, or multiple modalities, more than two even. And it finds a single k-nearest neighbor graph and a shared embedding, which you can then use to go on to do clustering and so on and so forth. So you can see that basically the flexibility of this approach means that you could integrate RNA with ATAC, with NM-seq, which is includes methylation with spatial data, even across species, because it allows you to take into account both the shared features, but also unshared features. So it leverages all features. So it allows you to look not just in the gene space of 20 or 25,000 human genes, but or 10 or 15,000 mouse. The features that are specific to one species or specific to one modality. So in ATAC sequencing, if you have 100,000 peaks, it will take all 100,000 peaks, and it will take all 20,000 genes, and it will consider the links between the peaks and the genes, but it will also consider the additional peaks that don't have a promoter gene counterpart. So I hope that's clear. It allows a nonlinear transformation, so it doesn't assume linearity. It's scalable because it's an efficient graph computation, and it finds relationships basically between the points. So the classical kind of computational biology, bioinformatics exercise on synthetic data, one of the kind of challenges is shown here, and where we're integrating the jelly roll, which is a three-dimensional coordinate system, with the rectangle, which is a two-dimensional coordinate system. And what MultiMAP finds is an embedding that's like a horseshoe that links the corresponding points to each other in the right solution, basically going from blue to red. And you can see here, there's a good mixing of the jelly roll with the rectangle. If we use the MNIST handwriting example for the digits 0, 1, 2, 3, 4, 5, 6, 7, 8, 9, you see 5 here. Each color is a different digit, 0 to 9. And what we're considering here is just 2 voxels in the middle, on the left and the right hand side. And basically MultiMAP successfully finds the points from the number 5, taking into account just the data set on the left with only 2 shared, and the data set on the right. And we can basically vary the extent of sharing to embed to see how successfully MultiMAP kind of finds the correspondence. Now moving to biological data sets, to real life biological data sets, we'll come on to synthetic data later, or gold standard data. We used a mouse splenic transcriptomic and chromatin accessibility data from my lab that we published a few years ago by Xu Chen. And in this publication, we showed the different cell populations of the mouse spleen here by single cell ATAC sequencing, and you can see different B cell populations, T cell populations, myeloid cell populations. And in the B cell compartment, there are 3 different cell states: marginal zone, transitional, and follicular. Now, if we take RNA sequencing data for the mouse spleen that we hadn't published at this time, then what we find is an additional B cell population that's an interferon responsive population, a 4th B cell population here. So there's follicular, transitional, marginal, and interferon, and that's basically found both in the RNA and the ATAC sequencing through, but we couldn't see it from the ATAC only because there were simply too few cells. And taking the 2 together, we can tease out this additional cell state. And so here you can see a different way of looking at the joint clustering is also the mapping here, showing how the RNA basically links to the RNA cluster, links to different cells from 2 clusters in the ATAC. And here's the new population. The other thing that I want to highlight is that MultiMAP allows, it sharpens clusters, and it also finds technical clusters. So with UMAP, for instance, doublets are distributed across different cell populations, but MultiMAP basically recognizes the technical features and pulls them out. So it kind of sharpens the clustering. And it's the other thing I want to highlight is that it's useful from a visualization point of view. So by running this exercise on the mouse splenic transcriptomes and epigenomes, we discovered new B cell states. We can show that in this data set, you get sharper clusters with MultiMAP versus UMAP, and it illustrates the power of the visualization for highlighting also a technical artifact cluster. I also want to mention that MultiMAP can be used as a batch correction method. Now, it's not designed as a batch correction method, and it's not necessarily the best batch correction method, but it's very fast because it runs in this graph computation, and you can basically use a whole bunch of 10 data sets or 16 data sets, integrate them together. And this is an example of hematopoiesis data from a recent paper in Nature Biotech, where there are 16 data sets, both ATAC and RNA. If we consider just the 6 RNA data sets and the 10 ATAC data sets and compare UMAP and MultiMAP, you can get a visual impression of the basically the batch integration here as compared to here, and the clustering of the cell types, and basically for the ATAC, the comparison between the UMAP at the top and the MultiMAP at the bottom. So I don't want to advocate MultiMAP as a batch correction method, but I just want to point out or highlight that it's one possible application of this framework. So it's scalable to large data sets, many batches. It's flexible for both within and cross-modality integration. And if we now go back to the 2 modalities that I mentioned earlier, the RNA and the spatial data, I'm taking an example of mouse cortex here, so the mouse brain, the outer layer of the brain, and 2 different data sets, one in cell and one in science. So the RNA data set is about 20,000 genes, obviously sort of mammalian genome, and 70,000 cells, whereas the spatial data set is a multiplex imaging, which is only 1,000 genes. So it's a small subset of the transcriptome, and also a much smaller number of cells, 2,000 cells. So can it successfully integrate the spatial data with a small number of genes and a smaller number of cells with the transcriptomes on the full transcriptome and a much larger data set? And here you can see the imaging, the star map spatial transcriptomics data here in the red dots, kind of more sparse, the RNA data here in the blue dots, and MultiMAP is basically overlaying the data sets and finding the best correspondence between the spatial and the transcriptome. And again, here, one thing worth pointing out is that the RNA data has a more nuanced, or the RNA and the spatial data have different definitions of the layers of the brain, the layers of the cortex, and basically what this joint clustering allows, that takes into account both the RNA and the star map data, is a definition of astrocytes, basically in different of glial cells in different layers of the brain. And so it's basically allowing us to take the clusters from the transcriptome and assign them in more detail to their spatial localizations and the different positions in the brain. So again, there's a benefit from taking the data from these 2 distinct publications, integrating them with MultiMAP, and learning in more detail about the glial cell state, so the supporting cells that are working to support the neurons, and basically assign their positions in the cells, and also actually the neurons themselves as well. So you can see the glia and the interneurons. I mentioned before that MultiMAP gives a kind of sharpening of the clusters, and we can quantify that with the silhouette coefficient, which basically measures the within versus across, the coefficient of clustering of spatial localization of cells within a cluster compared to across clusters. And what you can see here is using the example of endothelial cells, which are the cells of the blood vessels lining the blood vessels. They're nicely clustered separately from the neurons and the glia, and have a higher silhouette coefficient than in the UMAP, which basically shows the the endothelial cells here, cluster 16, are basically just right within the neurons and the glia, which is, maybe not as accurate a representation of the biology. So it's just another supporting example of the utility of MultiMAP. Finally, another feature of MultiMAP that I mentioned earlier was that it's not just pairwise, but it's basically allows you to integrate up 3, 4, 5, however many different modalities you want. So up to now, I've just shown you RNA and ATAC, RNA and spatial, but here is an example where MultiMAP is integrating ATAC, methyl, methylation of the DNA and RNA. And you can see the modalities in red, green, and blue. And basically it's successfully, integrating all 3 different modalities, which, it's more flexible than the other methods in this respect. And going back to the benchmarking, basically you can see here that we've integrated the splenic example of the RNA and the ATAC, the cortex with the RNA and the spatial, and then the visual cortex and the primary cortex with the 3 modalities. And MultiMAP is the only method that really does reasonably well on all 3. The others kind of run out of memory because of scalability, or can only consider pairwise data sets. If we again do the benchmarking on those 3 data sets, then MultiMAP performs well in terms of transfer, label transfer, silhouette coefficient, which is this clustering, alignment, structural coefficient, and runtime. Basically, it has a fast. I've mentioned that before. And it also performs well with label transfer in all of these exercises. The silhouette coefficient, which is that clustering within clustering versus across clustering, is high. The alignment does very well, although SURAT 3 is doing better here in terms of the splenic alignment, but in the other examples, MultiMAP is doing well, and the runtime is fast, which you can also see on the graph here. Now the sort of gold standard in a biological sense, I showed you the toy examples of the jelly roll and the MNIST writing earlier, but the biological gold standard is where this data set of human blood, peripheral blood mononuclear cells, where we have both transcriptome and open chromatin from the same single cell. So we kind of know what the solution is for integration, and we can use this as a benchmarking, and we can compare MultiMAP to other methods, and again show that the label transfer, silhouette coefficient, it does best in terms of alignment, again, almost as good as SURAT 3, the structure of the data and the runtime are again the best compared to the other methods. So, and this is just the summary again of showing the clustering of RNA and ATAC on this gold standard multi-ohm data set. Of course, if we had all modalities on every single cell, we wouldn't need MultiMAP or any other integration method. But a lot of the time we don't have this, but this can provide basically a gold standard benchmark. I'm going to finish off now by showing you an example of the utility of on T cells, which are my favorite cell type. And T cells have are the cells in our body that directly recognize foreign peptides on other cells in terms of killer T cells, or they recognize foreign peptides that are presented by dendritic cells in the case of helper T cells. And both helper and killer T cells have a T cell receptor on their cell surface. It consists of 2 chains, alpha and beta, that you can see here. And there's a diversity in each of the chains that's generated in the thymus during VDJ recombination. And it's that diversity in the T cell receptor that allows us to recognize the plethora of different antigens, different foreign antigens that we're presented with. And of course, the T cells, the T cell receptor sequences that present self are filtered out inside the thymus during the development of T cells. Now, the thymus sits kind of right next to the heart, or stuck to the heart. It's a small organ that was discovered as an immunological organ in the 1960s by Jacques Miller, and it's been studied by the immunology community in a lot of depth since then. And there are still new things to discover, and that's where the power of single-cell genomics comes in. And we studied the T-cell development in the thymus and published a paper on this in Science in 2020, last year. And what we showed was the using RNA sequencing and VDJ sequencing for the T-cell receptor is the pathway of differentiation in a lot of detail, and also discovering new, actually, CD8 alpha T-cell subtypes. And you can see the hematopoietic stem cell comes from the bone marrow, enters the thymus, and then develops through these double negative, double positive early phases towards the CD4 helper T-cells and the CD8 killer T-cells, which you can also see on the right-hand side. And basically I won't go into this in detail, but the V and J segments of the alpha and beta chain of the T-cell receptor are recombined by an enzyme called RAG to create the diversity of the protein complex to recognize diverse pathogens. And what we showed in that earlier paper for this data set was the pseudotime analysis, recapitulating known markers and then identifying new markers and new transcription factors in these different single positive T-cell states. Now, since then, we've generated ATAC sequencing data. And so my challenge to Mika and Mariana was whether they can integrate the ATAC sequencing data with the RNA sequencing data. And I showed you those transcription factors, that transcription factor network that we defined for the development of the T-cells. And I wanted to see whether we can also understand what the enhancer opening patterns was during the T-cell development. And Mika and Mariana basically integrated the data with MultiMAP, defined an integrated pseudotime for both the ATAC and the RNA sequencing data. And you can show you can see the markers and how they're kind of coming up during pseudotime on the right-hand side throughout the different phases of T-cell development. And then what Mariana did was basically look for the pattern of expression of transcription factors throughout the pseudotime from the hematopoietic stem cell to the differentiated T cells, and also define the average open chromatin peak for all the transcription factor binding sites throughout development. And so there are some transcription factors where you can see the enhancer opening is basically tracking perfectly the transcription factor expression, or enhancer closing is tracking perfectly decrease in expression. So there's decrease, there's increase in expression. But then there are other cases where the enhancer and transcription factor binding site opening isn't tracking the expression perfectly, or there's complex relationships basically between the increase in the RNA of the transcription factor and the opening of its binding sites. And so this is kind of at a global level, giving us insight into transcription factor expression dynamics and enhancer, the dynamics of the opening of the enhancers. And that really takes me to the end of the story on MultiMAP. What I basically want to leave you with take-home messages that MultiMAP can integrate multiple different data sets on one single shared manifold and embed in a shared space. It can integrate a diverse range of single-cell multi-omics data that facilitates label transfer, joint clustering. It can be used for batch correction. It's very useful for visualization, and it can also be used for things like joint pseudotime inference that I just showed you. And in the thymocyte pseudotime that I just mentioned, it can quantify the transcription. It can be used for further analyses that look at transcription factor enhancer relationships. The resources are here, the bioRxiv version 2 manuscript, and then the GitHub link. I want to say thank you again to Mariana, Mika, and everybody else who was involved, and I'm happy to take questions. Thank you.
Okay, thanks a lot, Sarah, for this presentation with a lot of results and also this introduction to MultiMAP. So I think we have some questions in the chat. So, okay. Well, a question from me to the organization. Am I supposed to read the questions, or did the person want to directly interact and ask the question?
I think we can just keep the questions that we have in the Q&R, and then you can read them and share them with everyone and answer to the person who asked for it. Okay,
so let's start. We have some questions. So a question from Sylvain, who said that if you understood correctly, MultiMAP can perform a joint clustering on single-cell RNA and single-cell ATAC-seq data. So how does it deal with biological replicates?
So it can happily integrate replicates. And I showed, I don't know if you remember, but I showed that example of the human hematopoiesis datasets where there are 6 RNA datasets and 10 ATAC datasets that are effectively biological replicates. And it can basically integrate the RNA replicates with themselves, the ATAC replicates with themselves, or all 16 data sets at the same time. And then Sylvain is asking, Sylvain is also asking a second question, where he or she says, is there anything planned for differential analyses like case control studies or cell differentiation dynamics? Yeah, so the answer is it's not a differential abundance assessment framework. For that question, I would like to advertise Milo. Milo, M-I-L-O, is a differential abundance framework on a k-nearest neighbor graph that Emma Dan, who's a PhD student in my group and co-supervised by John Marioni, has on bioRxiv and on GitHub together with Michael Morgan. So please take a look at Milo for addressing the question that you're asking about the differential abundance analysis.
Okay, so the next question, perhaps we can take the question from Ushan. He wants to know, or she wants to know, whether you check that SERAT, the 4th version, with weighted nearest neighboring, whether you check this integration method from multimodalities too, and what was the result?
So our benchmarking, which was kind of done several months ago, I think was on SERAT 3, not SERAT 4. So we haven't checked that outcome. I mean, I think weighted nearest neighbor integration can be, weighting can make a difference, and actually also in MultiMAP we have weights for different data sets and different neighborhoods that can be adapted flexibly. So there is a parameter optimization that can make a difference. Thanks.
Okay, perhaps you can take the question from Matthias now. And actually, I had the same. So we see that MultiMAP is performing significantly faster than the other programs. Could you justify or elaborate or explain why is it so fast?
Yeah. So basically, it's not a matrix. Like the matrix factorization method, for instance, it works on in a graph space, and there's a pre-computation of the principal component analysis, basically to calculate PCs between the pairwise data sets, and it then basically finds the optimum in that in that pairwise space, and
And this
so I would say MultiMAP is faster and it scales better than, it scales better than all of the existing methods, but there's still optimizations that we can do further, actually. And I mean, given the rapid exponential kind of increase in data sets in all different modalities, I think for MultiMAP, we need to think about that, but also for the whole field. It is really important to think about scalability and speed. Yes, yeah.
Thanks a lot. We have a question from someone anonymous, but the question is interesting. So, terrific talk, thanks. Could you please elaborate on how the gene expression and the regions defined by the ATAC-seq are actually associated by MultiMAP?
It's just very simple, it's just promoter peaks. So it's the peak in the promoter of the ATAC is linked to the expression of that gene. And so what it means is that, let's say if you have 100,000 ATAC peaks and, or, let's say, I don't know, 30,000 ATAC peaks, but you've got 2,000 genes expressed in that cell, then the 2,000 genes, will be linked to the promoter peak in the ATAC. And so you'll have some shared feature space, but then you'll have the other peaks that are not linked to the RNA.
Okay.
I hope that makes sense.
We have 2 questions left, one from Eve. So thank you, Sarah, for this very interesting talk. My question is more related to single-cell proteomics. Do you think that mass spectrometry-based technologies are ripe enough to allow for spatial proteomics?
I mean, certainly the codex type, the heavy metal linked antibody panels, are single cell resolution and they're reasonably multiplex, like 50-plex or so. So that's one, one approach. So what do you think that MS-based technologies? Yeah, so I think that, yes, certainly those data sets are very complementary to the, spatial transcriptomics data, because they're telling you about protein levels in single cells in a 2-dimensional space. And so certainly, MultiMAP or other methods can be used to integrate those modalities with the spatial transcriptomics, single cell transcriptomics, and so on. Yeah, for sure.
Okay. And the last question, perhaps, what is the next step for spatial transcriptomics? What about Merscope from VisGen?
So I think basically spatial transcriptomics technologies are an incredibly exciting area of technology development, and there are next steps on many different fronts. So one is, higher resolution, like slide-seq kind of single cell, or, going from 55 micron in the Visium to 5 micron, let's say. And not having dead space between the voxels. So I don't know if you saw that there was always like the 15 micron or so dead space. So having high density, high resolution, full transcriptome tissue section data, is incredibly exciting. The MERFISH Merscope from VisGen is a another orthogonal approach based on imaging. That has a plexity of, I guess, a few hundred genes, but very high resolution and even subcellular resolution. And that can be incredibly exciting and valuable. For instance, when you have cells that are very long or have complicated shapes like neurons, and you can define exactly where each individual molecule of RNA is located inside the cell. The previous question that we had about proteomics-based spatial transcriptomics is another future step, for defining proteins in space. protein expression in space and single cells. So I think the whole area has a lot of next steps in different directions that are really exciting. And from a computational point of view, you know, it gives us new challenges to deal with those data sets, to process, to analyze, to integrate. So it's a fantastic time for computational biology, I think, in the cell atlas domain.
Okay, so thanks a lot. It was a really nice introductory talk for this JOBIM edition. And really thank you, Sarah. Perhaps everybody can thank you also, I'm sure. And
thank you, Emmanuel. Thank you for
yes.
inviting me. It was a pleasure.
Thanks a lot.